good evening for someone, and welcome to the virtual biodiversity data use workshop. And this year we received like 138 applicants, and finally selected 40 because of the limited seats. So all of you here are like among the hundreds that representing 17 countries across Asia. So that's really nice this year. And before the session starts, like let me introduce myself and also let my partners introduce themselves later. So I'm Melissa Liu from Taiwan, and together with Lily Strata and Vijay Brave from the GBIF Asia Regional Support Team, I have also been working at the GBIF node in Taiwan since 2018. It's known as like type if. Okay, so look, we will be like also leading you through the next 2 days on how to use GBIF mediated data for biodiversity research. And we hope you can learn from the great resources as we delve into the course content. Okay, so I'll just let my partner to introduce. Maybe Lily first.
Hi everyone, good morning, good afternoon. My name is Lily Shrestha and I'm from Nepal. I'm also working as a GVIF Asia Regional Support Team together with my colleagues here, Melissa and Vijay, since last 4 years, more than 4 years now. And the major role that we have, major task that we have in this role is to help any data publisher or data user from Asia region in publishing their data, or if they want to use their data, if they have any questions around it, we act as a help desk for Asia region. Apart from this, I was previously working with the International Center for Integrated Mountain Development, but for now, I'm just working in this role as a GBIF Asia Regional Support Team. So please feel free to contact me if you have any questions around GBIF data mobilization and use. Looking forward to working with you in this two days Zoom session. Thank you.
And VJ.
Hello, my name is Vijay Barve. I'm from India. I'm currently based in Nashik in Western India. I I've been working with GBIF in different capacities for a long time, but last 4 years as a uh, Asia support team member. I have background in computer science, but I've been doing biodiversity informatics for a very long time. So that's briefly my background, and I'm happy to be part of this team and uh, leading this workshop.
Okay. And yeah, and also I think like some of you might already know this because like some of you like attended data mobilization workshop before also. Okay, and so before we introduce the activities these 2 days, I like to cover some like housekeeping and ground rules. Like first, the Zoom session will be recorded, and they will be available for all of you, all the participants after the workshop. And when you sign up for the workshop, we ask you to complete the media release. If you did not agree to the media release, then you should please like keep your camera off so that we are not recording your image. And if you post on social media, please use the hashtag GBIF Asia and tag GBIF. And do not share any photos of people. Screenshots of materials or presentations is fine. We will do like Zoom selfies today, later, and tomorrow. And that photo can be shared as people who don't want their image shared can opt out. So it is good to keep also to keep your microphone muted to minimize background noise during full like group sessions. And you can use the chat function and to ask questions or contribute additional information. You can also use the reaction button to raise your hand if you like to be called during discussions. So today we will not have breakout sessions and will mostly discuss all the exercises in the main room. But tomorrow we may have breakout discussions depending on the attendance. Okay, so please like unmute your microphone and participate during the discussion sessions. This is your time to ask questions. And if you need to miss a session or part of a session, please also let us know in advance. Okay, and this is today and tomorrow's the detail of the session. So today we will have like two sections. And the first section is to introduce introduction to GBIF using GBIF mediated data, and we will have exercise one about the Darwin core, and then is the gbif.org search functionality, and we will have exercise two about the gbif.org exploring, and then we will have like group photo, the selfie I just mentioned, and we will have 15 minutes break. And to section two, we will have like data quality issues and flags presentation. And then the exercise three is like examine download issues flags. And the last will be Q&A. And for tomorrow's session, uh, we will have like session three, part one is like GBIF API. And exercise four is like finding taxon keys, and then QA and introduction to RGBIF. So then we will also take another group photo, like to have before and after photos. So and 15 minutes break. Then the last section is all about the exercise like RGBIF. So there will be a demonstration how to use RGBIF to analyze and download the data, and then discussion, Q&A. Okay. So we will go to the first one, the presentation, Introduction to GBIF. So it's like I'll give the floor to Lily.
Thank you, Melissa. Let me start by sharing my screen. You can see my slides, right?
Yes.
Okay. So hi again, everyone. Again, Lily Shrestha here from Nepal. So I have also been sending you course emails in the last week. Hope you have got it. I know you have got it because you are here today. So I've mentioned in my email on Monday that you need to complete Introduction to GBIF course as a prerequisite. So I hope you have all done that. So the first part of my presentation will be just a brief review of what you have learned in Introduction to GBIF, but not focusing on data mobilization for sure, because this is workshop on data use. But I will also be combining the using GBIF mediated data in the same presentation, and we can have discussion afterwards. So in the first part of presentation, few slides introducing you to GBIF, and then we'll look into how GBIF mediated data can be used or is being used, and what are the general principles that we need to think about when we are using GBIF mediated data. So let me start with what GBIF is. We all know that GBIF stands for the Global Biodiversity Information Facility, and this is an international network and biodiversity data infrastructure that is funded by the countries that are participant to GBIF, the voting participants. GBIF aims to provide anyone, anywhere, free and open access to data about all types of life on Earth. So that's GBIF's main mission. But along with that, nowadays GBIF is also focusing on how GBIF's mediated data is being used for research and conservation actions. GBIF formally speak, GBIF works through voluntary. GBIF is a voluntary collaboration, sorry, that works through memorandum of understanding that is agreed between the participant countries and GBIF Secretariat, which is the core team, and the Secretariat is located in Copenhagen, Denmark. There are also regional support teams that is helping GBIF, assist data publishers and data holders in the region, and we are one of them from Asia. So this is the screenshot that you can see in the screen of gbif.org. We will be exploring about gbif.org in detail in the coming sessions. But just to let you know, the numbers that you can see on the bottom is the richness of biodiversity data that has been made.
Lily,
Yes,
I still see your first slide only, the title slide.
really? Is that the same?
Now,
Now
just
go
now
to the
it's
second
updated.
one.
Can you
Yes.
Yeah,
s- can you let me know if the slide changes now when I'm
yeah,
changing?
yeah. Now it is changing.
Okay.
Yes.
Okay.
Now you are on what is GB.
Okay. Sorry, I didn't know that my slide was not changing. So it was what I was presenting about earlier, just an introduction of GBIF. So I was saying that the numbers that you see on the bottom is the richness of biodiversity data that has been made available in GBIF so far. You can see more than 3.6 billion occurrence records, and that's huge number of record, right? Of species has been shared in more than 121,000 data sets. And these occurrence records and data sets are published by around 2,600 publishing institutions from all around the world. As I just mentioned before, GBIF also tracks the use of GBIF mediated data. So altogether, 14,358 peer-reviewed papers have been found to use GBIF mediated data. Did my slide change?
Your slide
No.
was stuck on species occurrence record.
Okay, what's happening? Let me stop share
Yeah,
and share it again, I guess.
Or maybe you should click on the screen, then it will change.
Sorry?
You can click on the screen on the screen you share, and then
Mhm.
it will change, I think.
Let me try again. I never had this problem before. Let me know if it changes this time. Did it?
yeah,
Yes.
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 Third slide. Okay. I hope it will change smoothly now. The occurrence records that I was talking about, more than 3.6 billion, has also multimedia
Previous slide,
evidence.
please. Previous slide.
Oh, okay.
No.
Yes.
Oh,
We did not
Correct.
get to see this at all, so
Oh,
that's the reason I
yeah.
oh
should
my
said go to
God.
the previous one because you talked about all these numbers and we didn't see them at all.
Do you want me to quickly summarize this
No,
slide
that's.
again?
I think that should be fine. Now we can see the numbers in the bottom.
Yeah,
So,
so the number that I was talking about, more than 3.6 billion occurrence records, 121 data sets, 2,600 around publishers and publishing institutions, and the data has been used in more than 14,000 peer-reviewed papers. But don't worry, because we'll be seeing this homepage a lot. The third slide, it is about the multimedia evidence associated with the occurrence record that I was just talking about. So there are around 267 million records that has taxonomically identified images, with 1.8 million odd, I mean, along with that, 1.8 million audio files and around 5,478 records with videos. So this kind of multimedia evidence can be very helpful for evidence-based reuse of data if you're concerned with that. The map here shows the network of data publishing institutions from all around the world. So there are altogether 148 countries or areas with institutions that have already started sharing data through GBIF, with the top being United States, Colombia, United Kingdom, Australia, and so on. Just to give you a brief overview of how data is shared, how data is made available in GBIF, and how one can use the data. So if you look at this diagram from the left hand side, the sources of data that comes into GBIF is from natural history museums, government agencies and ministries, field science such as NGOs and INGOs and other research organizations, citizen science network, business organizations, and academics or universities. They record their biodiversity either in the form of specimens, mostly concerning to natural history museum collections, field observations from the observations can also come from literatures, also from sensor-based instruments such as camera trap and remote sensing, and more commonly these days, the environmental DNA sampling. Once you have these biodiversity occurrence records, you have to standardize it because GBIF use a standard that's called Darwin Core. I will also be explaining a bit more about it in the coming slide. And we have three Creative Commons licenses that you can choose to publish your dataset with. I will also be talking about that later. So once you have your data set ready, you can publish it from various sources, but one of the most commonly used platform is called Integrated Publishing Toolkit. You can upload your data, you can publish and register with GBIF, and once your data is registered, it will be available, GBIF will index your records and it will be available for download from cloud services, gbif.org, and APIs. And then you can obviously use it for any of your research purpose or policy making around it. So that's generally how GBIF data is coming into GBIF and how it is being made available. Also, if you have gone through Introduction to GBIF course, it has been explained in detail, but just to let you know quickly, because it is also important for you to understand the levels of data supported by GBIF when you are working, when you are trying to use the data. So starting with the first one, that is the dataset metadata. It is a simple dataset that can be shared with GBIF. So it will contain description about your dataset that may be taxonomic, geographic, or temporal, but it will not contain the actual record of the species. It is just dataset metadata. This type of data set can be published, can be useful to be published. For instance, when your occurrence records are not ready to be published, or your natural history collections are not digitized yet, but you still want the users to know that the data exists and or you will be soon publishing the data sets. Then comes next three level of data set that actually contains the record of species. The first one is species checklist. We all know what checklist is. It is just a list of species within any region or concerning any thematic area such as invasive species or medicinal plants. So these only contains the list of species within any geographic area. And then the third and the most common type of data set is called occurrence data set. Apart from the list of species, as in checklist, it will also contain information on where the species was observed, the geographic coordinates or locality, and then when the species was observed, also the date of observation. So that's the main core of occurrence record. Along with the list of species, you will have date and coordinates and location information associated with it. And then finally, we have sampling event dataset. This is the most complex form of data that can be shared in GBIF so far. It will also contain occurrence records of the species, but along with that, you will also have an additional file describing about the sampling methodology that has been used. For example, the sampling protocol name, effort, and so on. So that's the difference between all that type of data sets shared through GBIF. I already mentioned before in GBIF, we use a standard and that's called Darwin Core Standard. It is a list of fields and their definition relating to biodiversity data. And this is used as a common standard for sharing data in GBIF because I've already mentioned there are like 2,600 publishers from 148 countries and areas, right? So if they start sharing the data in their own format, the data can be misunderstood. So to have a common understanding of the data that is being shared, we use this standard. It provides a simple way to document and share information about species occurrences and can help you answer your research and policy questions. So this is what Darwin Core is about, and there is a quick reference guide you can refer to if you are unsure of what term is being used in GBIF to know what they are and what are the examples of Darwin Core terms. The slide here shows the use of GBIF mediated data that GBIF has been tracking. Since 2008, you can see a steady increase. So the numbers has only been increasing. The data is from up until only September 2025. That's what GBIF had provided so far. So I have only this data, but obviously we have higher numbers for 2025 as well. So this was just quick introduction to GBIF based on GBIF's introduction to GBIF course. Now the next section of my presentation, I will be talking a little bit about using GBIF mediated data, how data is being used or how it can be used, and what are the things that we need to consider when we are using GBIF data. Starting with how GBIF data is being used, this figure is an outcome of a systematic review of the use of GBIF mediated data by Heverling et al. 2020. And in this study, a comprehensive analysis and review was done of more than 4,000 peer-reviewed studies that was published between 2003 and 2019 that relied on data from GBIF network. So you can see the data, the figure clearly shows that the data use from GBIF is interdisciplinary, mostly used in biology, but also in other fields such as earth sciences, medicine, chemistry, and so on. The studies showed both data availability and data use have increased over time, which is also true up until now for GBIF data. And usage of GBIF mediated data span disciplinary boundaries and conceptually diverse, which is already shown in this figure here. Now, let me talk a little about how GBIF mediated data could be used to support and address global policy needs on biodiversity. And the most important framework to look at, obviously, when we are talking about biodiversity, is the Kunming-Montreal Global Biodiversity Framework that was agreed during COP15 in 2022. So you can see in this figure how GBIF data relates to the targets of global biodiversity framework. For instance, if you look at target 1 to 8 set for reducing threats to biodiversity, GBIF provides primary data on species occurrence, which is required for target 1 on identifying key biodiversity areas, target 2 on monitoring restoration of ecosystem, target 3 on protected areas, target 4 on species conservation, target 6 on invasive species management, and target 8 on modeling climate change impact on biodiversity. Similarly, when we look at other targets, target 15 on assessing risks from biodiversity, GBIF does provide platform for sharing EIA-related data, biodiversity assessment-related data as well. And lastly, when we look at target 20 and 21, this is what GBIF actually does. When we are talking about capacity building, and this is what we are doing today as well, trying to build capacity on data use. We also run data mobilization workshops, trying to build capacity on data mobilization or how data can be shared. And lastly, target 21 for making best data available. I've already mentioned that's the mission of GBIF. GBIF exists to make species data freely and openly available for evidence-based conservation actions and decision making. That was about the Global Biodiversity Framework. Now, when we look at another big global policy framework, Sustainable Goals, GBIF also supports quite a broad range of SDGs. The most easy to look at are directly related, such as SDG 14 and 15, Life Below Water and Life on Land. And GBIF mediated data directly supports the conservation of both life on land and life below water by providing primary occurrence records of the species. Other SDGs we can look at that GBIF could contribute to are SDG 2 hunger, zero hunger, by providing data on agricultural species, fisheries, and many aspects of food production. SDG 3, good health and well-being. The data available through GBIF supports policy goals relating to human health, especially regarding to vector-borne diseases. And lastly, in relation to SDG 13 climate action, GBIF supports global efforts both to mitigate and adapt to climate change by providing biodiversity data for modeling impacts on biodiversity. So that's how GBIF data can also be used to address the sustainable development goals. Some of the examples in studies where GBIF mediated data can be used is the ecological niche modeling. We all know ecological niche modeling predicts suitable habitat for a certain species based on the data of where the species has been currently found or observed. And this model correlates occurrence data from species native range. And one of the source for that could be GBIF data. The occurrence data correlates with environmental data provided at a given resolution, and the suitable habitat for the species is predicted. So species occurrence data should actually reflect the precision of the environmental data that is being used in this model, and higher the number of data points, the better the model fits. So this is an example of where GBIF mediated data can be used, and obviously it is being used in ecological niche modeling studies. Next example is red list assessment of IUCN, which assesses the extinction risks of a species against a set of objective criteria. They have different criterias, but mainly criteria B on restricted geographic range. To calculate restricted geographic range, two of the metrics is required, that is extent of occurrence and area of occupancy. And for both of these, GBIF, georeferenced locality data and GBIF mediated data can be used to calculate that and then access whether the species has restricted geographic range, whether they are threatened to extinction or not. That's another example. So after letting you know a few examples of where GBIF mediated data can support policy needs, as well as how GBIF mediated data can be used for studies, I will now go into the access points to GBIF mediated data, how you can access the data. There are two common points. The first, most easy access to data is through gbif.org for people who are not into programming, and that offers advanced search,
Lily,
visualization. Sorry, yes?
the slides are stuck again. You are still on GBIF's contribution to sustainable development goals.
Really? You should have told me earlier. Is it changing now? I don't know what's
No,
happening.
Did you click on it?
Sorry?
Did you click on it? No.
Okay, let me share again.
You just like click. Maybe it's good.
I don't know what's happening. Try and share again. Is it changing now?
Yes.
Okay. This slide was where I was talking about ecological niche modeling. This is, I think, comparatively easy for us to understand because we are all from the same field, and also about red list assessment. It's changing, right?
yes, you are on the red list now. Now you are on access.
Yes.
Okay, sorry about this. I don't know what's wrong with my computer today. So yeah, I was talking about the access point to GBIF mediated data. And the most common and easy access to data is through gbif.org that I just mentioned for people who are not into programming. And that also offers advanced searching, analytics, visualization, and downloads for registered user in 12 different languages now. Next, we have a more programmatic way of accessing data through GBIF API, and that can be used to make advanced queries that are not supported by website. One of the ways of accessing GBIF mediated data through GBIF API is by using R, and we have a dedicated package called RGBIF. Don't worry, we will be learning about that in detail tomorrow, and we also have an exercise on how to access GBIF data using R. In the last part of my presentation, I will be talking about what are the general principles or golden rules to be considered when we are using GBIF mediated data. And these are the 5 points that I want to talk about, but I will also be explaining that in the next slides. So let's skip this for now. The first and the basic step is to set up an account in gbif.org. You can obviously explore the website without an account, but if you want to download the data, then you need an account. It's not only for downloading the data, but you can having an account, you can also get information on all the downloads that you have performed in GBIF in the past, or any derived data set that you may have. So that can be very useful, and using a dedicated email or password pair is recommended because it will also help you in the programmatic ways of accessing data when you provide the email and password there to access the data. So yes, you must have an account on gbif.org. Second, I've mentioned that GBIF data is free and open to use, but I would also like to say that it is not free of responsibilities. So every users must agree to the GBIF data user agreement. This is a non-binding agreement between users and GBIF, and it sets out the rules by which users should be using the data. For example, due attribution of the use of data through correct citation practices, using the data according to the license with which the data publisher has made it available. I'll be talking about the license just now after this slide. And also notifying GBIF regarding any data quality issues or other issues in their website that you may have found using the feedback mechanisms provided by GBIF Secretariat. So as a user, you should carefully read the data user agreement before you use the data. Did my slide change?
Yes, the license.
Okay. So I talked about license a couple of times. This is what I meant. So in GBIF, when the data publisher is sharing their data, they share their data using three of the Creative Commons licenses. The first one being CCO, for making the data available for any use without any restrictions. CC BY for making your data available for any use, but it mentions that appropriate attribution is to be given to the data publisher. And lastly, CC BY NC, where data can be used for only non-commercial use and with appropriate attribution. So these are the three licenses that every data set, every data records in GBIF has, and you should be careful when we are using the data about which licenses the data are associated with. Obviously, with when we are using the data, GBIF data does not. I mean, there are some issues associated with GBIF data for sure. We will be looking at that also later in the session today. We have a dedicated session on looking into data quality issues, and by knowing what kind of data quality issues might exist within the data, we hope that you will be comfortable in making decisions for your research. But after downloading the data or when you are using gbif.org, there is always some post-processing of the data that we do. This will require an approach that will balance the requirement of your analysis and also the robustness of your analysis. But it is always recommended that you document how you process your data. We are saying this because GBIF is always growing, the data in GBIF is always growing. So if you are doing this analysis today, data processing today, and if you want to do that again in the few months' time, can be useful when you when you document the process by which you are processing your data, because GBIF data can change over time. Now the slide is not changing for me. Okay. um, Next is about the formats by which you can download your data set. Data can be downloaded in 4 different formats now. A simple download, which contains a smaller file that contains smaller number of interpreted data from GBIF, but not raw data, and no multimedia if there is any. Darwin Core Archive that contains all of the original data and GBIF interpretations and also includes link to multimedia. So if you need all, Darwin Core Archive is the one that you are supposed to download. Species list is just a list of species names found within the data set. And a relatively new addition is the cube download. This download format allows you to aggregate the occurrences by their, for instance, taxonomic, temporal, or spatial properties. That can be very useful when you are doing modeling such as species distribution modeling, and the result is a CSV file. So these are the 4 formats of download that you have in gbif.org. When I'm talking about raw data and interpreted data in my next session on exploring gbif.org, I'll be talking about that as well. Lastly, after you download the data, please correctly cite the data that you use, however you are using it. Every data downloads are issued with digital object identifiers, DOI, that you are encouraged to use when you cite the use of our data, along with the correct citation practice. And the literature tracking system in GBIF can detect the use of DOIs from online libraries such as Google Scholar, Scopus, Wiley, Springerlink, and so on. And that will allow us to show how data provided by the data publisher is being used. So citation practice varies depending on how you're using data from GBIF, and you can get a full overview of correct citation practice from the first link given here on citation guidelines. And there is also a new development that's called derived data set. For instance, when someone is using data from different data sets, but they are also doing some post processing, removing some records, and so on, and they need a new DOI for the combination of data set that they have used, they can use this tool to let GBIF know which data sets are being used, which records, and also provide a backup online version of the processed data set. Then GBIF will provide you a unique DOI for the data set. So it's always possible for you to generate a new DOI for the combination of data set that you are using through derived data set tool. And when you are downloading the data, the citations for the data are provided to you in the page. After you download the data, you are redirected to a new page, and that shows the citation, as well as you will also get the citation in your confirmation email, the email that you have registered in gbif.org with. You'll get that, get the citation there as well. So keep the citation handy so that you can use it when you have your research paper published. Lastly, once you have processed the data and used it for your analysis, please deposit your processed data in a public repository, such as example could be Zenodo, with the associated download DOI from GBIF, so that anyone can access your data in the future. That's the last one. And if you remember the golden rules of GBIF mediated data about having an account, I will just show you the page, I guess. Yes, having an account on gbif.org, agreeing to data user agreement, documenting how you process your data, citing the data, the most important, and depositing the used data in a public repository. So with that, I would like to end my long presentation. And sorry for the interruption in between because of the slide problem. Thank you for listening to me. And Melissa, if you have any time for discussion, or we are taking discussion towards the end, it's up to you. Over to you.
I think we may have the QA session for the last.
Okay,
Yeah.
sure.
And okay. So, because of the time, now we just go to the exercise one immediately. And I hope before that, okay, before I share my screen, I want you all just make sure you already download all the files you need in this workshop. It's like Libby already shared the training course before, and please like go to files for download, and there are like some all the exercise sheets and videos. So please like download the exercise sheet, at least like exercise one for now, because we are going to do it. Okay, and then let me share my screen. Okay, so I think you can sh see my screen, right?
Yes.
Yes.
Okay. So it's exercise one, and we are like we will go for like exploring the darn core terms. Um, okay. And like when you get your data, your spreadsheet will like contain a large number of terms that you may or may not be familiar with. And the data available is like often much richer than you think. So in this exercise, we will introduce you to a small slice of the total number of terms available to you. Imagine it is the first time. you have seen Darwin core terms. If it is not your first time, just imagine. Okay. And you would like to do a study using GBIF mediated data to characterize a species habitat and determine population sizes. So you can also choose a species that you are familiar with and just like please like view the Darwin core terms on your exercise sheets table. So just like please think about that. Which of the following terms would provide relevant data for you for your study? So the terms are individual count, occurrence status, associated media, associated taxa, organism ID, material sample ID, event date, habitat sampling protocol, and minimum elevation in meters, decimal latitude, type status property, vernacular name, and recorded by. So I'll give you about 15 minutes to think about like which terms you should use in your study and why. So and then after 15 minutes, I'll pick up someone or maybe some volunteers to say about like which terms you use. Okay, and which term are you don't think you need. Okay, and I'll set up it's not the session break. It's okay. That's okay. Let's come back. Yeah, I hope everyone is already complete the sheet. And could you please tell me? Okay, let me maybe share my screen again. Wait a minute. Okay. Okay. Can anyone be the volunteer first to say like what terms you use? Okay, I see Ashak. Raise your hand. Okay.
Can I do it?
Yes, please, Ashak.
Yeah, I'm sharing. Can you allow me to share the screen?
Yes.
Entire screen. Entire screen. Yeah, my screen visible?
Yes.
1 second. What has happened? Okay. So, uh, so I was since I am more into occurrence and event data sets, not dealing with anything related to collection of specimens, and I am thinking about no relevance of these Darwin Core terms in case of an event or an occurrence activity. And the terms I'm using, individual count, it is important and it is required since we are trying to assess a species restricted to a geographical range. And we can, using this particular field, we can figure out the rate at which this particular species is either declining or it is kind of it is either gaining this presence or it is declining. And sometimes the assumptions may occur in counting a species, and if you the species are seen more in number or somebody wants to give something which is fictitious, this individual counts may sometimes go wrong. And occurrence status is also important, and it is actually a mandatory field in a occurrence data set. And so it has to be there if you are going for an occurrence data set. This occurrence status is important. Associated media is important since it is going to authenticate whether the species is present or not. So if you're looking for a species which is under threat, or it is there is some issues with its red listing, and it's gone beyond that. So this media is very important to ascertain its presence. And the problem is that somebody may use identify a particular species wrongly. It also is a situation where you have use associated media that is not giving the correct identification, or it is not the species which you are basically observing. It can be a different photograph altogether. That is the challenge of the data which we see. Associated taxa is not important, and we are in my case it is not important because we are looking for a target species. So associated taxa may not be that relevant for our case. Even organism ID is also not necessary because we are working with some target species, and always it is not always available. Organism IDs are not available, and we may not be use this in our data set. And material sample ID is not required because we are not working with samples, and we may ignore this field. Event date is important, and because without that we cannot create an event or an occurrence data set. So, and if you that's the problem is if you wrongly enter a event date, that may create problem later while using the data. Habitat is not always required because if you are working on a known habitat, and for a restricted species, this habitat details are not always necessary. To my sampling protocol is always important because if you are using an event, and you are using some Pollard or some other methods of the protocol used for your creating those data sets, it's important, and we may use it. And elevation in minimal elevation is not necessary for, in my case, it may be required for other cases, and often it is not measurable if you are not having the proper gadgets with you. And latitude and of course longitude also, they both are important to ascertain the presence of the particular occurrence in a field. And accurate measurements may not be possible. That's why there is a field for tolerance also along with the latitude and longitude. So including these three, we can have a assessment of the location of particular taxa. And type status properties also not required in the occurrence datasets. Vernacular names are required are not required if you are targeting a known species, and people who are doing this work are trained enough. So this also is not required. Recorded by is important because it will ascertain the authenticity of the record and who has genuinely recorded this particular species. And I think these are the fields required for my in my case. Thank you.
Thank you, Ashak. It's like you're really like familiar with Darwin core terms and know like which to use and which are not. And, like some of most of your like explanation are really great. And but some of them are a bit like interesting. I just want to know if there is like someone have different answers. So maybe Aditi, you want to share yours?
Yeah, Alyssa. Should I share my screen?
Yes, please.
Yeah. Can you see my screen?
Yes.
Yeah, actually, honestly felt 15 minutes were very less to give right answers. But I'll try to add my things. Most of the things I agree to what Ashok sir said. But where I feel that a different, what I have opinion is, he said associated taxa may not be required. But I don't know, when we are talking about conservation, there can be sometimes situations where some data can get easily skipped out, and if we are including the associated taxa, maybe that will not be filtered out. So if we are missing, that is what I feel. And apart from that, I do not have much difference in what he had said. I have additions to what I think the further things which might be required asset. Like basis of record was not mentioned. I think basis of record is one of the
I
crucial
could not
occurrence.
complete it. I could not complete it.
Yeah. So that is one thing. And then since we are talking about conservation, organism quantity is relevant. I feel it is relevant. Then sex may also be relevant because if we are talking about some particular taxa where gender and gender ratio affects the conservation status or something like that, so the involvement or including sex might be important. Same is the case in case of life stage. So we do not know. In certain cases, life stages may be important or maybe uh, they can to be important in designing and driving the conservation strategies. I don't know, maybe. Very sure. Location array is important, which was not there, which is crucial when we are talking about conservation status. For this related to it, same way the geodetic datum and identified by or identified by ID. Because if identification is not correct, maybe the whole thing can go. So this is what I could another 15 minutes.
Yeah, thank you, Aditi. That's excellent. And yeah, I'm also curious that yeah, life stage is like I think for some species they are really important, but it depends
Yeah.
on like if you can recognize or identify their life stage of
Yes.
some species. Yeah, but some species really difficult to know about that.
Yeah.
So
I
also
was
depends.
more thinking when I am saying life stages, I was more thinking on in this direction that sometimes if we are talking about conservation status and if we are thinking on those lines, what are the crucial factors which are causing the organism to be considered under conservation status? That is what I was thinking. I don't know whether I'm able to express myself clearly or not. I mean, the selection pressures are different for different life stages.
Mm-hmm.
So if something related to that is that there is a particular kind of negative selection pressure on a particular life stage. So if we can control that, the whole thing can be improved. The whole status of the organism can be. In that reference, I was thinking that maybe life stage can be.
Yeah,
I don't
for
know whether it was clear to what. I mean, whether I was able to articulate what I am thinking. I'm
yes.
not sure.
VJ, you want to add something?
Yeah, regarding the associated taxa, that is the place where you can write about host plant of a insect or thing. So it is important in case of conservation when you are looking at their biotic associations and stuff. Otherwise, in many places, it may not be available or recorded. So that's what I wanted to add. And also, we need to discuss about organism ID and material
Yes.
sample ID, the question that was raised. So,
I would like to add to what he is saying that it's very true. When since we are talking about conservation, there will be a lot of things which will be important apart from the organism details. Because when the organism exists in the environment, it is associated with so many other organisms which can play crucial role in its status. So I do agree with what just now. And you want me to discuss which two? Material sample ID and
And yeah, organism ID, I can maybe I can share one experience. Like if you can, it also depends on the species you choose. If you can like recognize like each individual. For my example is like one of my friends study about the sea turtle and they do the photo identification, so they can like track individuals of the sea turtles, and they will also use the organism ID. So that would be important for them to know to track like every individual of the sea turtles, like which about their maybe habitat or maybe to do the analysis. So uh Yeah, so it also depends on it because like not every species can track with the individual things. And if you have any other like experience example, you can also share.
In case of tigers, I think they do the same thing.
Yeah.
Yeah.
Yeah,
Okay.
tigers and some elephants are actually named in India, so they know
Yeah.
which
Yeah.
particular individual is moving in which regions and stuff like that. In case of the material sample ID, it's basically to link to the original specimen. Sometimes what happens is when you in your collection you have a specimen, but you remove the tissue and store it separately for molecular work. In case of birds, suppose you are making preserving the skin, sometimes the skull or other bones are separated out and they are preserved separately to study. So these are the kind of typical uses of material samples. Each of these samples are attached with ID so that they can be backlinked to the specimen and ultimately to the individual in some sense.
Yeah,
I think, yeah.
yeah, I just take my research for example. I do the stranded cetacean, dolphins and whales. So I will take their organ and tissues as my like material sample. So that ID will help me to track back to which like observation we have. Okay.
Dr.
Yeah,
Shoaib
same
has
is
his
case
hand
in
up for some time. Yes,
may
sorry, go
I
ahead,
add up
Aditi.
something? Yeah.
Yeah,
Yeah, the same thing is there when I am working on mods. I do the same thing that we have the ID and we connect it with the barcodes and everything. But for the conservation thing, what we have given for the exercise, I don't know whether it is really mandatory because generally wherever the conservation status is in question, generally the collection is restricted. Hence, I feel that it might. I don't know whether it would be relevant.
yeah. Right.
That's it. Should I stop
Yeah.
sharing my screen?
Yeah, yeah.
Yeah, you can
Doc,
stop sharing.
Dr. Shoaib has his hand up for some time.
And just answer one of the sorry, it's like Roy's question is like material sample ID only meant for the museum specimen? No, not really. Because like not every even it's not a collection. It will maybe your research will also need a material sample of your species. So just the example I mentioned before, my research, I'm not working in at the museum, but I will collect the sample from cetacean. So it's not only for museum specimens. Yeah. And like for if someone is doing research about the eDNA or DNA metabarcoding thing, they will also need a material sample. So yeah, that's not only for that purpose, collection purpose. Okay. Okay. Any other questions?
And Dr. Shoaib has his
Uh,
hand up.
uh,
Yeah.
thank you, Vijay and Melissa, for giving me some time to share. I'm unable to share the screen regarding the Darwin, the core terms, but I've been as a student of a botany and the field collector and having some background of spatio-temporal assessment of the species, I would like, rather I tried to rearrange the number of the terms according to my studies. And for me is one should have to note down the event date, and it would be followed by the who is recording that data, and that should be coupled with the elevation in meters and along with the altitude and latitude, specifically with respect to the distribution of the species. as I was working with some international agencies few weeks back, and we were working on a distribution of a species, particularly with respect to the association of the plants with the avian fauna, and in which the habitat description is very much important as well. And followed by the sampling protocols, and then the checklists or the nomenclatures in case of plants, which is very much important. And particularly, all the species in the fields are not usually identified in case of plants. And in that particular case, one has to give some ID in order to work on it in a herbarium or in a museum or something like that. And specifically for the vernacular names is concerned, in the field, when we are going to perform such kind of a sampling or some kind of a collections, the vernacular names are very much important with respect to ethnobotanical knowledge, with respect to the knowledge used by the indigenous people of a certain area. So vernacular name is very much important. And if we have to go for the quantification of the data, the individual count is can be considered with variable parameters. And the occurrence in terms of number should be followed this. And particularly in plant studies, and specifically in order to describe the habitat, the associated taxas are very much important to describe the community or the habitat composition. And for me, the material sample ID, if you are going to take some other samples, having some fruit samples from the area, the material sample IDs are very much important. And it should be later on linked with the associated media. I hope so. You wouldn't mind it. I rather change the sequence of the terms with respect to my studies and the collection. Thank you so very much.
Thank you so much. It's like I just want to like make the conclusion that it's like not there are not like only correct one the answer of this exercise is always like depends on what kind of species you're search research for. And yeah, so it's like it's interesting. Like we can hear from different answers from different field of research from the researchers. So that's why we need to have this kind of discussions because that everyone has different point of view. Okay. And no only one answers. Yeah, it's
Thank,
more than yeah.
thank you.
Thank you. So because of the time, we need to go to the gbif.org exploring. So just like Lily, it's your time for presentation again.
Thank you, Melissa. Um, so for this part on searching on gbif.org, we will be exploring how we can look into data from GBIF's website. Obviously, we have a part on API and R tomorrow, but just going through the website, how searches can be done, how it can be filtered to refine your search, and so on. So let me start by sharing my screen. I hope we don't get that problem again. But anyway, uh, so so this is the homepage of gbif.org that I also showed you earlier. Um, and here are how you can access different data, like from the occurrences, all the 3.6 billion occurrences that has been published from the occurrence tab, data on the species in GBIF backbone taxonomy, the data sets that has been published, the publishers that have published these data, and any resources that GBIF has made available to you. So here in this live demo, I will be dealing with first looking into a species and how a species space looks like. Then we'll go into occurrence search and apply some filters there. We also have an exercise following on the occurrence filtering and search. And we will also look into the spatial search in occurrences. So that's what my demo now will be basically focused on. So let's start from the species search. We can directly go to the species tab, or we can also do a free text search here. So let's say I'm looking for a species called Asian elephant that's native to the Indian subcontinent and Southeast Asia. So let's just do a free text search here. Don't worry when you see this security verification in order to prevent attacks. GBIF has done this now. So it might take a little bit time for it to respond, but it happens with everyone. So when I did a free text search, Asian elephant, it shows me everything the word is in, including the data sets, the publisher, species, page, and so on. So because we are looking into species, let's go to the species tab. I'm just doing it around because you could also go through the species page itself. Now, again, we have 11 results. And let's say I'm not sure about the scientific name. Then what we can do is we can apply some of the filters here. So in the rank, I will say that I'm looking for a species. In the status, I can say I'm looking for an accepted name. And in higher taxon, I will say that I'm looking for mammal, right? So these are the three filter that I applied, and it decreased my option to 3 results. Obviously, I'm not looking for carnivores. I'm looking for elephants. So, and I'm also not looking for primates, macaques. So this is my the species that I'm looking for, Elephas maximus. And this is how a species space will look like in gbif.org. In the left hand side, you can see the higher taxonomy of the species, including the synonyms of the accepted names, also the subspecies information. A subspecies can also be found in this particular tab called for and fras species, and you will find individual pages for each of the subspecies there. Now let's start from the top. We can see the species is accepted and its scientific name with scientific name authorship. The name when it was first published and described in System Manager in 10th edition, and it says that it is in GBIF backbone taxonomy. Asian elephant in English, this is the vernacular name, and there is trade restrictions associated with Asian elephant in CITES, as we all know. So this is what the top description is about. And in the overview page, you can see we can see that there are 11,034 occurrences associated with this particular species, among which 2,220 occurrences have images. So if you want to look at only occurrence with image, you can go to See Gallery and look into the occurrences. Next section, out of 11,034 occurrences, 5,325 are georeference records. So in the map, obviously, only the georeference records can be shown. Interestingly, we can see, although the species is native to this region, we can see some of the occurrences points there and in Europe and also in North America. So let's look into that later on when we are dealing with the occurrences. Next section is on description of the species, including any information on breeding, habitat, ecology, their activity, conservation status, and so on. And all the sections has individual sources for you to refer to. It might not be available in detail for all of the species, but you can just look into that as well. Next section is on IUCN status, saying the species is endangered. And then the vernacular names, different common names of Asian elephant in different languages. This is about the data sets where Asian elephants occurrences or records are in. So it says that it appears in 32 checklist data sets and 112 occurrence data sets. So if you want to see what are the occurrences in this particular data set, you can click on these occurrences and just look into that. And lastly, very important, I have already mentioned the citation. This is how you should cite if you are using any information from the species page in gbif.org. Let me quickly scroll back to the top. And there is also another section here on treatments, taxonomic treatments. Just note that this section might not be available for all the species, but this particular species has the taxonomic description and history of Asian elephant, along starting from when it was described to others, along with the sources as well. So this is all about species space. Simple. You will just need to look at what sections are available and what information can be useful to you when you're looking at the species. This is about species search. Now let's go into occurrence search. I'm just going to give a demo of the same species. The occurrence is published in GBIF of the same species. So I can directly go from this link here. But what I can do is the most common way for us when we go from the home page is clicking on this occurrence tab and then applying the filter in scientific name saying Elephas maximus. Okay, this one, right? So we just select, you can see there might also be, when you are doing this filter with scientific name, there might also be synonyms that are being shown. So you just need to make sure that you use the correct name in order for the occurrences to be shown correctly. So this is the scientific name I want to look into the occurrences of. This is the occurrence search interface. You can refine your search based on your research questions. There are different filters that's available in simple filters. If the filters are not available here, the one that you're concerned about, you can also have a look in the all filter section. So I'd just like to quickly go through the simple filter section. GBIF always applies this automatic filter on occurrence status present, but please note that GBIF also has records on absent. So if you want to include that as well, you can say everything. Next is the license that we just talked about in my earlier presentation. So just make sure you use the data according to the license that they are assigned with. For instance, if you are doing a study for some commercial use, then you need to skip these data 4,260 occurrences with non-commercial, that says prohibits the use for non-commercial use. So that's what we should think about when we are doing that. So you can apply the filter here. Scientific name, obviously I have applied the filter. Basis of record is the next session I would like to quickly let you know about, since it's important for us to know. I know many of you have already published data in GBIF and you're well aware of this term, but for those of you who are not aware, basis of record is the nature of your species observation. How have you observed the species? What is the nature of the species observation that you have in GBIF? So you can see there are three set of observation, three set of specimen and sample material and material citation. So let's start with the observation. Sometimes people just don't know the criteria and they just apply observation and occurrence. These are the very simple terms. But please note that basis of record is a controlled vocabulary. And when you are providing the data, when the data publisher is providing the data, they can't just write any word in that, and they have to choose among this vocabularies. So the machine observation, as the name says it, observed with the help of machines such as camera trap or remote sensing. Human observation, it says that we have directly observed it in the field. Material sample, some materials that we just described about, part of the organism, DNA, and so on. Material citation, that comes from literature. Observation that comes from literature. So the literature has already been published and you derive the occurrences from that. Preserved specimen, obviously the name says it, it's from the natural history collections, preserved specimen. It's also fossil specimen, also usually comes from natural history collections, but fossils. This is just a way of distinguishing fossils and other specimens. And living specimen from botanical garden or zoo. So this is what basis of record is all about when you're trying to use the data. You can also apply filters on the temporal scope of the occurrences here. If you want to say you want data from 2000 to 2026, then you can do that here. Month and the location. I will quickly go through location in next step, but let's see what are the filters under that. Location, administrative areas, and country or area. I will just it will follow. So the next filter that I'm talking about obviously is continent. You can apply filters here if you just want to say you want records from Asia. Then the data sets, there are different data sets that the occurrence results has been published in. You can select that. Also the publishers that have published these data sets and records. IUCN Global Red List category. Obviously, most the records are endangered. And lastly, the important one that I wanted to quickly discuss is the issues and flags. So I mentioned earlier that not all data in GBIF is free of free of issues. So you need to make sure you look into what kind of issues or flags are there with the data that you are concerned about and that you want to download and use. It shows what record, how many record had what kind of issues. So you just need to make sure you look into that. We have a dedicated session afterwards on data quality and issues that will be led by Melissa. So we can look into that later on. But just letting you know that there is this section where you can look into the issues and flags in gbif.org itself. I forgot to mention this in the beginning of the demo. I was planning to say this, but I somehow forget. So I let you know now because this is something with issues and flags that I want to show how it differs. Actually, the interface of gbif.org that I'm showing you right now, this occurrence interface mainly, it is going to change. The new GBIF website is coming in live, I think, towards the later half of the year. So I will quickly also take you through the demo website at the end. But for now, because GBIF has this website, we have to go through it. So the one thing that I would like to show you how it has improved is currently with issues and flags, you are not able to, for example, I want to, from these 11,034 results, I want to exclude the occurrences that has coordinate rounded issues. This is not a very important issue, but just giving an example here. It's possible to see the records with these issues, but it is not possible to exclude this in this current setting of the website, which is now possible in the new website I will show you later on. So issues and flags, and yes, let's go to the spatial search, how it can be applied, because mostly when we are looking at species, we are concerned with species at a particular location or a particular country or administrative areas, and these three sections are very important when we are doing that. So let's look into location first. In location, you can say whether you want only results that includes coordinate, or you can say there's no preference or without coordinates. That's possible. You can see a little map here, which you can zoom in according to your need, and you can zoom into the location of your concern. You can draw a quick polygon, delineate the area, and say that you just want to see occurrences from this particular area, and just roughly delineating the boundary of Nepal to see what occurrences are there. I'm not being strict here, obviously, but you can be more strict on that, and it will refine my results to the occurrences of Elephas maximus in Nepal only. So that can something be useful to you. You can also provide the bounding coordinates of the location of your interest here. You can provide the geometry in WKT or GeoJSON format, for instance, from the GeoJSON or WKT file that you get from GIS save file. That's also possible. So this is all about location, how you can quickly refine your search of the location. Now, let's say I will do no preference right now. I will also like to show you the differences between administrative areas and country or areas. So administrative areas, global administrative areas, the country or area section has only country level selection that you can make, right? But in administrative areas, it is also possible for you to make selections in the lower level of country, for example, divisions, subdivisions within the country. That's possible in administrative areas. But I would like to show you the difference of administrative areas and country or area in the country level itself. So let's first say I want to see the records of this particular species in country India. It will show me some records. Okay, it's taking time. 2556 results from administrative India when I select it from administrative results. Now let's clear that and do India in country or area. It's a slightly higher result than that was in administrative areas, right? So the only records with coordinates are shown in administrative areas, and in country or area, if the publisher just provides the Darwin core field country as India without providing the coordinates, that occurrences are also included in that. So that's basically the difference between these two fields. And this is a tip because we are also doing that in exercise later on. Yes, so that's all it's all about. If you do not find any terms of your that you want to apply filters in, you can also look in all filters section. There are different classes and many different terms, but depends whether the publisher has provided data in those terms or not. Now, let's look into the tabs of the occurrence interface. First one is the table, simply showing you the data on occurrence of the species. You can select more terms from the three dots here. So there are many different terms that you can select to be shown on the table page. And then next is the gallery. In gallery, you can see the images associated with the occurrences, and obviously it has decreased because only 4604 occurrences has images associated with it. Map, again the number has increased because remember in map only the occurrences with coordinates will be shown. So yes, only the occurrences with coordinates, sorry. So yes, we also already saw in earlier species space that there were some interesting occurrences outside the region of our concern. We'll look into that just now. Taxonomy, higher taxonomy of the species that we are looking into. If there is two species, there will be different taxonomic information here. In the metrics tab, you can see the summary of occurrences of the species that we are looking into. Occurrences per month, occurrences per year, basis of record, per license, per issues, and per flags, and per dataset. This is the default way of showing you the summary of occurrences, but you can also customize it according to your research need. You can see the summary of occurrences accordingly. So for example, if I say I want to see a summary of occurrences according to basis of record and country or area, this table will be shown where they will show me different basis of record and different countries where the occurrences are from. So let's look into United States of America because obviously Elephas maximus is not naturally found there. So the most of the occurrences are from preserved specimen from Natural History Museum. So that's why the points are there in the North American part. A material sample, DNA sample as well. So it's obvious the organism is not found here from when we look into the basis of record of these occurrences. But let's look into interesting three occurrences that says human observation. When I click on three occurrences from human observation and go to the table, I will only be shown the three occurrences. Let's look into the first one and see what's happening there. Yes, so there's whenever you are looking at a particular record, you will be shown this issues, so you can also look into that. But let's quickly scroll down to the location and see, and it says that it's from Jew, right? So it should ideally have been the basis of record should ideally have been living specimen, but the publisher somehow just put it as observation, and that's something we should always look into when we are trying to access or download the data set. So now we know why the points were showing in that region and some interesting findings in that as well. And lastly, when you are ready, you have done the filters, you're okay with your result, you can download your data set a quick review, simple Darwin Core Archive, species list, and queue format that you can choose from. You will be shown all the issues and flags of the search that you have done here again. So look into that as well and do the download. When you download, remember you need to be logged in. Otherwise, this page will not be shown and they will ask you to log in first. Um, Yes, and remember, I also mentioned about interpreted data, and I wanted to show you what interpreted data and raw data is all about. Obviously, raw data exactly as data publisher has provided, and interpreted data somehow GBIF interprets it. I will show you an example here. In publisher, I'm going to choose India Biodiversity Portal, the occurrences that has been published from this institution, and go to the table and select the first occurrence to just show you what interpreted data is all about. So when we look at this record, we can see the original data has event date, and GBIF just interprets any of the Darwin core field associated with it from the data that has been provided, year, month, and day field, right? Similarly, the data publisher has only provided decimal latitude and decimal longitude in the field, but has not mentioned that this is from India. So based on the coordinates, we will just interpret that this record is from India, country code is IN, and the continent is Asia. So this is all the interpreted data is about. So when you're downloading the Darwin Core archive, you will get the original data as well as the interpreted data. But then with the species, the first option, simple option, you can just get the interpreted data from GBIF. So yes, that was all about species search, occurrence search, and spatial filter in gbif.org. Now I would like to quickly show you how our new website will look like. It's you can also see now at demo.gbif.org. You can see the homepage of gbif.org basically looks the same. Nothing much will be changed, but the main change that you will see is on the occurrence tab, and the filtering page or the search interface for occurrence will look completely different. So you can see the same tab, but there is an addition of related that will show you relative occurrences like for other species, and there is addition of dashboard. I will show you dashboard in a while, but now it's also possible to quickly apply scientific filter from the heading itself. And you can also sort the data according to, for example, if I want to sort from country or area, I can do that. You can also apply the filters here, like I did last time for Elephas maximus. You can just apply here, your country or area, and issues and flags is something I wanted to just discuss about, right? So now what's possible is, for example, if I don't want, let's choose, okay, I mean, significant issue that will that might hamper most of the studies. Country coordinate mismatch. I can select that and I can say exclude selected. So it will only give me the results without these occurrences. So that's what is possible now and can be very useful when you are you are trying to access data from gbif.org. And there are more fields, all the fields that were there in simple and all filters. And quickly want to show you dashboard because this can be a very interactive way of showing the summary of occurrences as compared to the current website we have. You can create your own dashboard. For instance, let's say I want to create a table. Then I want to create a map of these occurrences. I want to also see the basis of record of these occurrences. And then I want to see, let's say, publisher who has published this data set. So I can just customize the dashboard according to what I want. And if I make changes in one of the section, let's do what I did before, only see occurrences from Nepal. So once I do small changes in one of the section, it will change accordingly. So it can be very interactive, right? I make changes here and then the occurrences will be summarized. And this is something you can also share with people. So yeah, that's something new to look forward to in the new website. Um, I don't have much time to just go through in detail, but we will obviously, I think in our when we have next data use workshop, we'll have to deal with this new website because that will come into life. Um, yeah, so that's all I wanted to quickly show you, and just letting you know, you can also search for data sets. From this dataset page, you can type the dataset name if you know, or you can just select publisher and then see what kind of dataset they have published. And you can also see what publishers are registered in GBIF from your country. For instance, if I do Nepal, it will show me 17 results from our country, the publishers in GBIF. This is everything I had in the demo. Now we will do a quick exercise based on what I've just presented. You also need to explore gbif.org and apply some filters and get me some answers. Let me share another presentation. Looks like I don't have it open. Just give me a second. Yes, I do have it here. Okay, so this is the exercise that we are going to do now. Sorry, I have the answers slide open. So these are the searches that you need to do in gbif.org. Just go to gbif.org, select occurrences, and then go to the occurrence search interface and get me the answers on what are the total number of occurrences for Thailand, what percentage of the data do not allow for commercial use, remember the license, how many fossils are there in Thailand, how many records for the bee family Aphidae are there in Thailand, how many of the Aphidae records in Thailand have an altitude above 900 meter, and how many of the resulting Aphidae records with an altitude above 900 meter are georeferenced, that means they have coordinates, and how does the total number of occurrence change when you use GADM filter as compared to one when you are using country or area filter. So that's just a quick exercise we will be doing. Melissa, I don't remember, do we go into break before or after the exercise? After, right?
After.
And how much time we have for the exercise?
Uh, Actually, it's like 15 minutes, but
I
yeah,
don't think we'll
now
need
we have
15 minutes.
limited time now. So maybe 10 minutes of that.
Okay, okay. So are you all ready to do the exercise? Just give me a thumbs up. I can just keep my screen on for you to see the questions. Sorry. Okay. Let's take roughly about 10 minutes to do the all this.
We can make it interactive. People can just start typing the answers to the questions in the chat box.
No, we can do that as well.
That will be quicker.
Are we almost done? Should we start discussing or you need some more time? You can just speak up, anyone.
I think we can start discussing.
Okay. So, yes, I think my next slide will be the answers for sure. But let me know if yours differ from mine. I just did this in the morning, so I don't know if occurrences have changed by then. But then these are the results that I got while I was doing it in the morning. So, um, any differences that you get, or any specific questions that you want me to show on gbif.org, or I can quickly show everything as well.
Ma'am, please share the number 7, how to check the GADM
Okay.
filters.
Okay. Maybe we can just quickly, it will just take few minutes to just see everything, but then I will need to be reminded of the questions. Let me share my screen. Go into, okay, I have to open a new page. Occurrences. So the first question was, how many species from Thailand, right? So in the country or area, I will just
How
type
many
Thailand.
occurrences, madam?
Sorry, occurrences. Sorry. Yeah, I meant that. Just a slip of tongue. So I'll just apply filter in country or area as Thailand and it will give me the result. Next question was on what percentage do not allow for commercial use. You can just see in the license the number that prohibits the use of data for non-commercial use, and you can just convert into the percentage. 720,685. That's the second one. So how many fossil records are there in Thailand? You remember I talked about basis of record. So you can see there are 3,927 fossil specimens from Thailand. Then the next is on bee family Apidae in Thailand. So in scientific name, we can just type Apidae and not genus, because we the question says family. We can apply that filter and see 17,077 results from the family Apidae in Thailand. Altitude above 900 meter, it's not available in simple filter. You have to look at all filters and find the term called elevation. And in elevation, there's option where you can say no filter, you can drag it, but it's easier to just say it says above, and then you can just write in this zero saying 900. It will give you the result. So you can, if you want to apply that result still, you can add, and it will show you 1,922 results. Then I go back, and I hope that that question on Subhajit on question on altitude is clear. And with georeferenced altitude above 900 meter at georeference, I can simply see in this map tab, and it will show me 1,821 occurrences with coordinates that are georeferenced. That's the answer for 6. And for 7, how does the total occurrences in Thailand change as compared to when you are using country or area and GADM filters? So this is the result I get when I apply the filter in country or area. And let's look into the administrative areas. When I apply Thailand, it will be a lesser number than what it was, basically because it only has records that has coordinates associated with it. So that's basically all. It's very simple because it's just applying filters on the available terms there. I just need to look into it a few times. Any questions on that? Or anyone has a different?
Madam, apart from the coordinates, are there any other issues which are excluded in GADM?
No, basically that's the difference in country. Sometimes what happens is when data publisher is publishing their data set, they will provide the Darwin core field in country as India or some other country, and they will not provide the coordinates. So because all occurrences do not have coordinates, and it's not always possible to georeference, but the data can still be useful, right? So the occurrences that has country or area field, but no coordinates will be in country or area, and in GADM it will only have divisions, I mean the location with coordinates in it.
So, madam,
But the difference is obviously that in GADM you can also filter according to the smaller divisions of the country, for example, a
yes,
district
yes,
or any administrative
I
areas.
understand that. So, in order to check how many occurrences are georeferenced, so we can instead of checking the map, can we also check the GADM filter?
I think that should also be work. I haven't tried. But you can also, there's also another way of doing that in the location tab. If you remember, there was an option if you want to include coordinates or without coordinates.
Mm.
So that's also there. I think JDM also work because that is the major difference on that. You
Another
can
question,
just
like
try
for
and
India,
see.
like for India, a section of India, the Pakistan occupied Kashmir or the Azad Kashmir, as they say, that part is not under Indian administration, right? So, for GADM, is that part included under the Indian territory or not?
I'm not sure about that. Sorry, I haven't looked into that detail. You can just search on GADM and see where that is, but I do not have an idea on the policy
Because
which
as
you
far as on-field administration is concerned, that part is administered by Pakistan only.
Mhm. So maybe it's
So
under Pakistan, but I haven't had a.
maybe that part is excluded from.
Yeah, this is a problem with eBird data also from Pakistan. That the birders from Pakistan, when they report, that thing goes to in the records it showed in India. So this is a problem which is going on for long regarding eBird also.
Yeah. Uh, yeah, the discussion is always there, but we also have a dedicated discussion session towards the end. Let's see how much time we have for that. But now I would like to hand over to Melissa for rest of the session.
Yeah, thank you, Lily. And so now it's the selfie time, and then we will take a break. Yeah. Okay. Welcome back. I hope everyone is back. So if you're back now, please give me a thumbs up. Well, okay. Seems many of you are back now. Okay, then we just go to the next section. Minute. Okay. Okay. So it's next in this presentation, we will basically explore issues and flags of the filter features on gbif.org. We will also provide related scripts for you to try in our studio tomorrow. Okay. Before you use the GBIF mediated data, you should notice that your GBIF download will not always be perfect for that you want to do with it. So there are a few things you should be aware of. Is during the indexation process over the raw data, GBIF adds issues and flags to records with common data quality problems. This may contain useful information for you as a user to create fit-for-purpose datasets. So when you try to download the GBIF mediated data, you might only see the appearance of the datasets. So the deeper you dig or filter out, the better the data for use. So let's take a look on different types of data issues you might need to be aware of. Okay, first is geospatial issues. So when you go for occurrence search get data occurrences on GBIF, you will see the filters of the field location, and then you will see the default like there is a feature of like you can check or uncheck. They include records where coordinates are flagged and suspicious. So by default, GBIF removes common geospatial issues if you choose to have data with a location. So there are like many issues they will remove It's like zero coordinate, country coordinate mismatch, coordinate invalid, coordinate out of range. So for the zero coordinate, like GBIF removes this. It's like some publisher might not provide the correct coordinate or no coordinates, but they just like provide the 0, 0 instead. So if that happen, we call it like zero coordinates. It will like go to show as a null island on the map. So GBIF will remove it. And and GBIF removes records that do not match their country code. So they will like match with the coordinates, the longitude and latitude, and if it's not in that country, the boundary of country, it will say is like country coordinate mismatch. And on certain location, this is an example that may need to excluded in your analysis. So this is a record from California, and the coordinate uncertainty in meters is like 12,205. Okay, which is like too large. You can see it's too large to know whether the species is on land or in the ocean or located somewhere. Okay. So often you will like want to be sure that the coordinates give a certain location and are not really like 1,000 of kilometer away from where the organism was observed or collected. So we recommend not filtering out missing values. Things like publishers often leave them blank when the occurrence is very certain. So, like, wait, okay. There are a few fake values for coordinate uncertainty that you should be aware of too. These values are errors produced by geocoding software and do not represent real uncertainty values. Like in the case of 301, the uncertainty is often much, much greater than 301 and actually represents a country centroid. So country centroids are where the observations is pinned to center of country instead of being closer to where the animal, plant, or microbe was observed or recorded. So some publishers consider zero and null to be equivalent. So empty latitude and longitude end up being plotted along these like two lines. So you will see these two lines. Let's see. Like this. It's like easy to see the line at the equator if they provide let me sure, the latitude in zero. Okay. And country centroids. So like what I mentioned, a country centroid is a point that is considered the center of a country. So not defined by a polygon. And then we have like centroid attached to it. So why this occurs is when people are georeferencing what we often have when we have a specimen as we'll have typically textual information. So we may only have a country or a city or a text description. So if we have this, for instance, a record saying this species was found in Japan, for example. So they will use the country centroid of Japan to put coordinates to that data so that they may take the information on the label and produce coordinates based on country central, but not the place the species exactly found. So the tips is to use the distance from centroid in meters on a GBIF like occurrence search to filter for country centroids. So the R package coordinate cleaner can also be used for identifying and filtering for country centroids. You will learn more tomorrow. And then the gridded datasets. And gridded datasets are a known problem at GBIF. Many datasets have equally spaced points in a regular pattern. These datasets are usually systematic national surveys or data taken from some atlas. So georeference occurrences are snapped to a central point. So most publishers of gridded datasets actually fill in one of the following columns, like coordinate uncertainty in meters, coordinate precision, or footprint WKT. Okay, so filtering by these columns can be a good way to remove gridded datasets. And GBIF has an experimental API for identifying datasets which exhibit a certain amount of griddiness. And absent records. Okay. Okay. Um, Sometimes data publishers will include absent records where they verify that a species is not present. So most of users don't want these records. So that GBIF by default will remove that records. But actually, not many data publishers provide a current status like this. But GBIF basically automatically interpret this from the individual count. So you will see if it's present in the current status. It means they have numbers in the individual count. And by default, both presence and absence records are shown like this in the interface, are shown when you search records on GBIF. So absence records, again, confirm that a species was not found at the specific locality when that area was surveyed, and this information can be useful in. So for example, developing ecological niche models, you like this field might be helpful for you. If you are only interested in presence records, you can just like filter for that used on gbif.org. And there are other issues you have to filter yourself. The first is basis of record. And GBIF has like fossils and living specimens. So usually a plant inside a botanical garden or sometimes an animal in a zoo. And even though this can be a useful filter, data publishers do not always complete the basis of record field correctly, or there may be nuance in the data that may not be immediately obvious to a user. So you should always double check your data before use. Okay, and another thing is that, for example, if you go on the occurrences searching, and then you choose the human observation on the basis of record, you might see if you go to gallery, yeah, immediately, it will see some photos, and might be some photos that is obviously not human observation. Like this is maybe is a machine observation, or some of even are like specimens. And the second is establishment means. Okay. Establishment means is the statement about whether a Darwin Core term organism has been introduced to a given place and time through the direct or indirect activity of modern humans. So it's the process by which the biological individuals represented in the occurrence became established at the location. And just I noticed that it's control vocabulary. But unfortunately, it's not used very often. Okay, if you choose the establishment means on the gbif.org, it will give you like these control vocabulary such as native, reintroduce, native, indigenous, introduce, uncertain, and introduce, and vagrant. Okay, these are the options you can choose. And the third is time duration. We take year, for example. So GBIF has many museum records, and that might be older than what is desired for some studies. So you can just like choose the year and move the controller, the bar of that to set which the duration you want. Okay, and then now we are going for the exercise 2. Data quality and issues and flags. Sorry, it should be exercise 3. Just miswrote a little bit. Okay. So, okay, it's the continue exercise from exercise 2. Okay. And check issues and flags from the family Epidae, occurrences in Thailand, and download from previous exercise, check flags. So you can think about how would you use the flags to check the data. Okay, so there are like also the same, actually is the same filters. So Epiday occurrences present in Thailand and with coordinates above 900 meters elevation. Okay, so there are 2 questions. So I will give you about also 10 minutes, and then you should answer me these 2 questions. The first one is, would any of the issues cause you to do further filtering before downloading the data? And the second is, which issues would be important for you to resolve after downloading the data? Okay. Let's count down for... Okay, I hope everyone already have answered about this, and I hope the time is also enough for you to explore all the issues on your searching. And is there any volunteer again to answer the question? Okay. What are the issues you think is important and you want to filter and to resolve after you download the data? Please raise your hand. Okay, before anyone raise your hand, I can have like add something on. It's like when you see all the data issues here, you should notice that not every issues are really kind of data issues. It's just a reminder, like the continent derived from coordinates and country derived from coordinates. It's just the GBIF like will uh index that field automatically based on the field that publishers provide. Okay. Okay, Ding, please.
Yeah, I can share, but yeah. But the last question I'm not very sure. So
Okay.
one. Yeah, so I searched for it, and then the occurrence in the results are 1,922. And then when I went to the matrix tab, this is what I found for the flags, the occurrence per issues and flags. So these are the things. But then when I look at the download tab, this they prompt like what you said, they will prompt us again. These are the potential problems that we might have. So after looking through and depending on what's the research question or what we want to find out, I may, which usually I want to make sure that what the occurrence is accurate in terms of where they are found and when they are found. So I would filter things out like recorded date mismatch and the identified date is invalid. So this might not be correct. And then I try to check the other things. I think they usually are not a big issue for what I want to do. So I think these are what I would filter out. But then this part I'm not very sure. The last question, is it to check the final numbers are accurate, but it should be automatically filter out. So I'm not sure then. Yeah.
Okay. Yeah, it's like depends on you will filter it out before you download it, or you just like you just like choose to download it first and then to resolve it. Because sometimes it's might also like better, not really might also better. Sometimes if you check the detail of the issues, like which like date is invalid, maybe it's easy to know what's the issues in it. Maybe you can correct it yourself, but sometimes you may not just know the correct answer, so you can just choose to filter it out. Okay.
Okay.
And okay. And um any other of you want to share your answer? Okay, and about the geodetic datum invalid, if that issue showed on gbif.org, actually it will like automatically to interpret the data as WGS84. Is the same geodetic datum as you extract the coordinates from Google Map. So if the publisher didn't provide that field, uh, GBIF will assume the coordinate is WGS84. Okay. So yeah, seems like there's not many issues to discuss about. Almost like Ding already answered. So now is QA time for open for all the sessions today. Any questions?
My one question. Just now you were mentioning that some of the data are geo-referenced to country centroid, and for some of my work, we will still want to know like what species are found in a certain country. It'd be nice if I know where exactly they are found, like which part of the country, but sometimes the only data that we have from GBIF, I guess, will be from the country centroid. So what should we do with such data?
I think you can still like get this information like from which country in the other fields like country or country code or locality. So if or you can just like try to download all of them and when you do the analysis of the coordinates, you just remember to remove it. Okay, VJ.
Yeah, so what happens sometimes is with some of the older specimens which are sitting in a foreign repository, at times they are not aware of the locality or the old state names. So they just leave the record at country level and sometimes put a country centroid as the point. But if we look at other location fields like locality, county, state, province, and things like that, we are able to refine the geolocation. Obviously, if it is not recorded initially, we may not get a very accurate location, but sometimes we might get, depending on what geographical unit we are able to find out up to uh, 20-kilometer radius rather than few hundred-kilometer radius, which is a country centroid. So, depending on how rare that organism is and how much data is available, we can always try to refine it.
There's another issues I think you cannot filter it out on gbif.org, but you should also remove it manually. It's like sometimes the specimens from museum, the coordinates might not be correct one from the species where they found. Sometimes they will provide the coordinates location of the museum is instead of providing the correct one. So yeah, just want to remind everyone, you may still like even you just filter out all the geospatial issues on gbif.org, when you download them and try to analyze them, you still need to like check it manually. Okay. There's one questions. Okay. How does GBIF prevent the misuse of biodiversity data, particularly species occurrence data, that could potentially be used by poachers for illegal hunting? Okay, I think we discussed this before last year. Also, almost like every year there will be a participant like talk about these issues. But I forgot, like VJ or who
Um,
provided a
I
good
just
answer, but I
no,
cannot remember
I
the details.
just I just want to start with my opinion, and you guys can add. But I just want to say that since GBIF is free and open access to all, there's no way that GBIF can actually prevent the use of data in any way once the data is there. Because and if you are concerned about the use of sensitive information, for example, on sensitive species, we recommend the data publisher to be as vague with the locality as possible. There is a definite guide on how GBIF publishers can publish data on sensitive species. We can paste the link here for your reference. But I think it's entirely up to the publisher what kind of data they want to provide, how sensitive information they want to hide, or how open they want to be with the data. So my point of view is that GBIF cannot actually prevent the misuse of data because it's free and open for all. if you guys want to add anything on that.
Yeah, just like what Vijay said, there might be like local, so they might be familiar with like where to find these animals than searching on gbif.org actually. And about the misuse of biodiversity data, not only like this example, but sometimes some like company, if they want to do some how to say the what is that term? The environmental assessment. Yeah. And sometimes they will also misuse of the data. But like what Lily said, we cannot really prevent from it, but to try to how to say it, engage more with like this community and then to promote the importance about the data quality and the proper data use.
Yeah, I just added a link on that which deals with how to uh, kind of manage your sensitive species data. Like you publish the data, but just sufficient to let scientists know that you have it, but not let poachers know where it is, and then the scientists can get in touch with you and work with you to uh, use that data properly for scientific purpose and poachers will get it.
Sorry, is that me, my side, or everyone here like, about VJ is not that clear because it's breaking?
Yeah,
Oh,
me.
okay, VJ, your sound is a bit like breaking. Can you say it again?
Is it better now?
Yes, better.
Okay. So I just put a link in the chat which talks about how to deal with sensitive data publication. And basically it tells you how to prevent giving out too much details to the poachers, but tell the scientists that you have the information and they can get in touch with you to work with you to protect that species. So yeah, take a look at that document.
And there's another questions, maybe VJ to answer. Ding just asked, will we learn how to check georeference data in tomorrow's session? You're muted.
Uh,
Yeah,
yes, a
okay.
little bit. We will deal with some of the georeferencing issues and uh, how to be aware and careful about using that data when we are using it for any analysis. We are going to talk a little bit about that.
Okay. And is there any other questions? If no, it's time for wrapping up and like to remind you about tomorrow's session is GBIF API and R. So the important thing, most important thing is like to remember you already install the R Studio and open it successfully. Okay. So if you have issues uh, on installing our studio, please stay in the main room and VJ will help you to yeah to solve the problems. Okay. Okay. And then um oh the last one questions in the last second. If publishers will choose not to share the exact coordinates, how would it affect the quality of the data set? It's a good question. It's some species might be like endangered, so it's kind of sensitive data, and then the publisher will not prefer to publish it actually. So they will provide the coordinate uncertainty in meters in maybe instead like it's quite big of the value and it might also affect the quality of data set and it's like at least you know the species is there, but you cannot know the exact range of the species habitat. But VJ or Lily, do you
Um,
want to share anything?
Yeah, I just wanted to add that in the document that Vijay has provided, there's recommendation on how when you are altering the coordination, making it generalized for a big location or a country, you always have to provide that information in your data set somehow so that the users should know that this is not the exact coordinate, either by putting the coordinate. There is a field, Darwin core field, called coordinate uncertainty in meters, so the value will be very high for that certain kind of location for such sensitive species. And it's also recommended to provide some remarks saying the coordinate has been generalized, it's not the specific coordinate, so that the data user can always get back to you in case they need exact coordinates, and you are doing I mean using the data for a good purpose for your research or something like that. I think the data publisher would not have issue on providing you the exact coordinates. So that's how it works, that's how it's supposed to work. But again, it depends on how data publisher choose to publish their data set.
Yeah, and just the quick answer of the Sabita question is like GBIF will not automatically generalize the coordinates. Only publishers to decide to provide it or not. Because like the species in different countries, the endangered status is different. So yeah, we will not automatically do it. Harsh?
Yeah, so my question is regarding the coordinate uncertainty in meters. So, of course, it depends on the research question that what that the researcher is setting up the uncertainty in meters, and what criteria or how many kilometers the researcher want. But is there any, like, I'm unsure about it, and I just want to know that if there is any criteria provided by GBIF that minimum this much of the uncertainty the data user should be taking into consideration or something like that.
I
Um,
don't think there is any criteria
no.
set for that, right?
Yeah. Do you have any other experience? Okay.
As far as I know, I mean, there's no criteria set for how big of uncertainty you can, because we believe that every data you have is useful. Sometimes, as we already talked about it a couple of times before, sometimes a specimen, museum specimen, does not have any location except saying that the specimen was collected from Nepal or India. So in that case, the coordinate obviously will have a very large uncertainty in meters, and that data can still be useful for some people. So GBIF does not have any criteria set up for that.
Okay, thanks.
Just to add to that,
Sorry, VJ, your
for
sound is breaking.
Yeah,
still.
I think it's the internet connection.
Oh, okay.
Now
Yeah,
it's fine.
it's
Yeah.
fine.
But
Okay. Okay. So just to add to what Harsh was asking, like a few years ago, there was not a data set even at country level for distributions of all the butterflies of the world, and we worked on it and created, which was very useful. And now using that as baseline, a lot of people are actually building upon that. So that's the reason at whatever level the data is, it is always useful.
I'm sorry, but your voice was continuously breaking and we
yeah,
couldn't
well, maybe
decipher what.
VJ, you can text in chat box. Maybe it's better. Okay. And okay, this thanks and uh okay, and the last question is because of the time, so we will just answer the last question. Okay. Like if I want to make species checklist uh of particular taxa in one country, and then I use GBIF and filter the country or area and also the uh yeah, what is that?
GAD.
Oh, okay.
Global administrative areas that I
Yeah, well, I
actually
mean, yeah.
I actually do not get the question clearly. But you are trying to make a species checklist using GBIF mediated data, right? And you want to filter with either country or area or GADM. I don't know if you can apply both the filters at once. Should be okay. But I think it will give you all the data that GBIF has when you apply that filter. So because in country or area, as I mentioned, there will also be data without the coordinates. So it's better to use country or area because you are concerned with the country and not specific locations inside the country. So I think country or area filter would make more sense because you're looking to make a species checklist of the particular taxa. But again, just to clarify that if you're preparing a checklist, GBIF might not have all the data that you might require, and you might need to look into other sources as well.
Yep. Okay. Okay. So yeah, it's time to say goodbye to you. And then just be sure you're already installed our studio, and please stay if you have any issues of installation. Okay, see you tomorrow at the same time, and I hope you learned a lot today. Hope you. Bye bye.
Thank you, everyone.
Thank you, Lily and Melissa.
Thank you so very much
You.
for a wonderful session.
Thank you. And thank you for the great feedback and discussion today.
Thank you. Goodbye.
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 Lily or VJ, is that, I'm not sure which VJ you started the recording, right? I think you can stop it now.